Good morning, everyone. Starting with the questions. So, give examples of some viruses.
Coronavirus, adenovirus, rotavirus,
Okay.
rhinovirus,
So while you classify, I asked examples of viruses. So while you classify, how do you classify?
DNA, RNA virus,
Okay. So what are the diseases caused by these viruses? Diseases.
main,
According to system. According to the system, they involve. Okay. Today we will discuss about the lab diagnosis of viral infections. So can you hear me? Okay. Before starting this lab diagnosis, we have to know the infections caused by the different viruses. Yes? So, the viruses, they can cause the sporadic diseases like mumps. Yes? Some viruses, they are endemic. I know. Some viruses, they cause epidemic. Now, after the rainy season, the dengue will take rise. Yes? Then after pandemic, no? That is simply coronavirus. Yes. Okay. So, and the symptoms caused by these viruses, they are non-specific. Okay. They are variable, ever changing. They are non-specific. So, for that, we have to decide what actually. Most of the time, they are non-specific symptoms like headache, yes, common cold or body ache and fatigue. So you have to decide very carefully that what sample should be taken for the disease it causes. Okay, let's take the example of polio. What type of disease is caused?
poliomyelitis,
Loud. Neurological? Yes. So if there is neurological disease, so what will be the sample? CSF. Okay. Now let's poliovirus. Wait. Don't. This is not the topic. Exact topic. You will get it in each and every book. Listen. Okay. So polioviruses, they are transmitted by fecal oral route. Okay. Most commonly followed by respiratory droplets via inhalation or rarely by conjunctival contact. So, how now? What you will do? Yes? Now, next. Polioviruses may be recovered from the throat swab up to 3 weeks of illness, rectal swab or stool samples up to 12 weeks of illness, and the excretion in this stool it may be prolonged due to immunodeficient patients. Okay, so and viral isolation from CSF and blood is very rare. So you have to be very wise while collecting the sample, deciding upon what test to be performed. Okay, so now let us start. So, viral diagnostics in clinical laboratory, why it is important? Because most of the time, over 60% of all infections that are seen by the physician, and in future you are, yes, so they are due to viral infections. Okay, and it is important that the quality specimens, okay, quality of patient specimens and their transfer to the laboratory, it is very important part. Okay, now these are the overview of viral infections that are caused: encephalitis, meningitis, okay, pharyngitis, common cold, cardiovascular disease, hepatitis, skin infections, sexually transmitted infections, gastroenteritis, pneumonia, eye infections. So there are different types of infections caused by the different types of viruses. Okay. Now, so as I have told, the symptoms produced by these viruses, they are ever changing, they are variable, okay, non-specific. So, and in the past, the viral diagnostics, it was very difficult, because the techniques, they were not much developed, okay, and it was of not much important because this antiviral agents, antiviral drugs that were not developed at the time, the jati it was so time consuming. जति दिनसम्म अब त्यो भाइरल डायग्नोसिस गर्यो त्यति दिनसम्म या त पेसेन्ट निको भइहाल्थ्यो or they would give up, that's why it was not much important. But now the technology has developed, okay, there are different antiviral drugs, that's why it is very important to diagnose the viral diseases. Okay, so why we do viral diagnosis? To confirm etiology of diseases and to, yes, to give the antiviral drugs for those viruses for which it is available. Then after post exposure prophylaxis, post exposure prophylaxis has a केमा दिन्छ. Have you heard about rabies? Yes. Okay. So have you heard about needle stick injury? Yes. So in those needle stick injury, while doing any work in the lab or in healthcare settings, about you don't know the status of patient, whether the patient is infectious or not, whether the patient has a blood-borne viruses, hepatitis, HIV, they are transmitted through blood. So we don't know the status of patient, whether they are infectious or not. So if you get needle stick injury, you should know the status of the patient. You should confirm the disease, patient कुछ हो कि छैन, and according to that you can give the post-exposure prophylaxis to prevent the disease. Okay. Then after is screening of blood. अब अस्ति भर्खर blood donation भएको थियो, कसले गर्यो. So before donation, we screen the blood, donor's blood, to prevent blood-borne infections. Okay, and that is most common the hepatitis and HIV. Okay, to monitor, detect, and predict epidemics and pandemics of viral infections earlier, so that appropriate control measures can be initiated. Okay. Then another one is to define the viral disease process. A disease process, this immunogenesis, pathogenesis, the symptoms they produce. This को लागि पनि त हामीले viral disease को virus को cultivation गर्ने पर्यो. Okay. So now, so for the diagnosis, the samples should be appropriate. Okay. That means the collection of the specimens for viral testing. For respiratory tract infections, it may be nasal and bronchial washings, throat and nasal swab, sputum. Also, if there is eye infection, conjunctival swab and scraping. Gastrointestinal infections were stool and rectal swabs. Vesicular rash. If there is a skin infection, you can take the skin scraping or ulcer scraping. Okay, and from vesicular fluid, you can aspirate the fluid by the help of syringe. Then after in CNS, if it is encephalitis or meningitis, then CSF, brain biopsy. Of course, brain biopsy it is only taken in after the autopsy. Then after throat swab, rectal swab, this is alibakar hamle polio ko example पढ़े है ना. Another one is genital infections, that is if there is vesicles formed, vesical fluid or hypogeneal swab or vaginal swab. Then after in urinary tract infections, urine, and blood-borne infections, blood. Okay, so wow, if you suspect of disease and need of the sample collection, it should be collected as early as possible. Okay, because there is several time of shedding of viruses in different specimens. Okay, so early collection it should be done. Then after collection, label the specimens adequately. Okay, with the patient's name, age, sex. If you know, you can write their provisional diagnosis, their identification number. So then after, use a small leak-proof specimen container. Okay, and use secondary protection against leakage, and refrigerate all specimens until tested. Right? So for bacteriological testing, you can keep the specimen at room temperature, according to the need. This is CSF sample. If you need to do for bacterial test, then you have to keep it in at room temperature. Okay, or in incubator. Incubator देखेंगे है ना. So incubator. But for all the viral testing, you have to keep all the specimen in the fridge, refrigerator. Okay, because the viruses are heat labile. Okay, now so excretion exudates, it may be feces, urine, saliva, sputum, nasopharyngeal aspirates, and CSF, tissue and swabs, and they should be transported in viral transport medium. कोरोना को बेलामा हेरेको थियो नि हैन, nasopharyngeal swab हैन, throat swab लिएर तिनीहरूले के गर्थ्यो भने एउटा मिडियममा राख्थ्यो हैन, viral transport medium. So to keep the viruses viable. Okay, so they should be kept in the viral transport medium. It prevents the specimen from drying out, prevents bacteria and fungal growth, and they keep the viruses viable. So this is the viral transport medium. सबैले कोरोना भाइरस को बेलामा एक्सपिरियन्स गर्यो होला हैन. Have you seen this one? Okay, so collection of stool, obtained as early as possible. It should be kept in a screw capped container, and several grams of formed stool or 5 ml of liquid stool should be collected, and for best result, collection within 2 days of illness. Urine. So 10 ml in adults, 2 to 3 ml in newborn and infants, and collected in leak-proof container. तिमीहरूले हेरेको छौ urine collection गर्ने. Okay, so refrigerate immediately, and if you centrifuge it, then the chance of isolation increases. Respiratory tract, upper respiratory specimen, that is nasopharyngeal swab, and oropharyngeal. It is more sensitive, more appropriate than the oropharyngeal swab, and it should be transmitted in VTM. Lower respiratory tract, जहिले पनि sample lab diagnosis हुँदाखेरि हैन, you have to start with the sample collection. Okay, and you should while writing in the answer, always remember that whether you are writing about the upper respiratory tract or lower respiratory tract, sputum is the sample always in the lower respiratory tract. infection. Okay, so and specimens that are better than this sputum are the bronchoalveolar lavas, transtracheal aspirates, and transbronchial biopsies. Skin and mucous membrane region, vesical fluid, and swab. CSF sterile screw cap tube, and medium is not needed, but instead of CSF, blood is not acceptable. Okay, and for blood and serum. Now cervical specimen, swab after removing the mucus and transport it in VTM. Semen collected in screw cap container. Eye specimens, they are collected by corneal scraping or simply taken the swab. So how we approach to the lab diagnosis? So for that we have to detect the virus in the specimen, okay, direct detection, okay, or we have to culture it for isolation of the virus or perform the different serological test, okay. So for direct detection of virus we can do by using electron microscope, electron microscopy, light microscopy for detection of histological appearance. This light microscopy is used after staining, when we grow the viruses in different cell culture, then for the detection of viruses, whether it has grown or not, there are different effects there, okay, in the cell that are seen, and the growth of virus, they are confirmed by staining and seeing under the light microscope, okay. So there are different histological changes in the cells after growth of the virus, and they are detected by light microscopy. Molecular methods, virus culture and isolation, they are done by animal inoculation, embryonic egg inoculation and cell culture, and serology is performed on the principle of immunochromatography and ELISA. So, direct detection of the effect. Originally, it was done by electron microscopy and inclusion bodies by light microscopy. Okay, but recently, immunoelectron microscopy is done. What we do is that when we mix the virus, when we mix the sample with antiserum of the specific virus, then it forms the agglutination, okay, and that are easily detected by microscopy. Okay, so that can be easily detected by, if there is clump formation, okay, then that can be easily detected by microscopy. So that is called immune electron microscopy. Then detection of viral nucleic acids by different molecular method, that is PCR, reverse transcriptase PCR, we will discuss later. Now electron microscopy. Who discovered electron microscopy? Resta. Yes? Yes? Loud. So it increases the magnification several times, okay, and diagnosis of viral gastroenteritis by detecting viruses in species, that is rotavirus, adenovirus, astrovirus, novavirus, they all causes gastroenteritis, okay. So these viruses can be detected in the stool by electron microscopy, okay. Now this, but the sensitivity of electron microscope is not much, that's why we modified it for immune electron microscopy, okay, and at least 10 to the power 5 to 6 viruses per ml should be, per ml of sample should be present for visualization. So this is the electron microscope, तुम्हें recording होला है ना, different types of microscopes class labs वाला है ना, okay. So now immune electron microscopy, I have already said the sensitivity and specificity of electron microscope is enhanced by immune electron microscope. How? Virus specific antibody. Suppose if you suspected of this, I have told no, rotavirus. Okay, so when you uh, mix the specific antibody against that rotavirus, okay, viruses, they are antigen. I know viruses for particles or rules and structure. You know what is antigen? Sorry, immunology पढ़ाई बात है. Okay. So when you mix the specific antibody against that virus in the sample, they will form agglutination. Okay, they will form clumps. Okay, and that will be easily recognized under the microscope. That is immune electron microscope. Okay. Now another one is the immunofluorescence. Okay. So when you tag the antibody with fluorescing agent, okay, what is the function of this fluorescence? They will convert the UV light into normal light so that it can be visualized. Right? So this fluorescent tag antibody, when you mix with the sample and see under microscope, then it appears like this. Okay, so that becomes easy. Now another one is the molecular method. Okay, so the non-cultural techniques. What is China? Okay, later on non-cultural techniques now we will discuss, learn in detail about the molecular methods. Okay, the principle, what are the types, how it works, what are the steps, we will learn it in detail. Okay, so the molecular methods, that is the PCR, polymerase chain reactions, reverse transcriptase PCR, real-time PCR, they allow amplification of the specific target. Okay, that is the DNA and or RNA sequence. Okay, and they are extremely sensitive and specific diagnostic assays. Quantitative reverse transcriptase PCR, they determine the viral load in blood sample for monitoring response. Okay, when you give the antiviral drugs, whether it is working or not, the viruses are responding or not, so that can be detected by this method, and example is HIV treatment and follow-up. Okay. So in situ hybridization using nucleic acid probe, that is done for human papilloma virus. So this is the process. The amplification is done. Okay, the DNA or RNA, they are amplified and detected. Now cultivation of virus. So viruses are intracellular parasite. I know. Have you seen the culture media here? So name some culture media. Culture media. Jago mohon pare jago. Come on. Wait. Name some culture media. चुप पानी देखे तो पातलो टेस्ट करना. So these viruses, they are obligate intracellular pathogen, I know. So they cannot be grown in the or this culture medium. Okay, so they need cell culture. Okay, so can't be grown in any inanimate culture medium. There are three methods employed for viral culture. That is animal inoculation, embryonic eggs inoculation, and cell or tissue culture. Okay, so if you see the history, if you go through the history, there are some evidence that the humans, they voluntarily have tested for the viruses. Okay, but nowadays it is not done. So now they use the animals for inoculation, and most common is the infant ones, that is suckling ones, rabbit, monkey, and if there is growth, whether the growth is there or not, that is indicated either by the death of the animal, or the diseases produced, or the visible reason. Okay, so another one is the chikungunya embryogenetic egg inoculation. You can see the different parts. So this is amniotic cavity. Okay, this is abdomen. This is allantoic cavity, yolk sac, chorioallantoic membrane. So there, the viruses, the different viruses, they are inoculated in different parts of the egg. Okay, so herpes virus, pox virus, sarcoma virus, they are inoculated in chorioallantoic membrane. Chorioallantoic membrane. Okay. Now, influenza virus, mumps virus, they are inoculated in amniotic. Please see here, amniotic. What is this? This is amniotic cavity. Okay, so they are inoculated in amniotic cavity. Also, yolk sac inoculation. Herpes virus is inoculated in yolk sac. Also in the chorioallantoic membrane. Okay, also influenza virus, mumps, Newcastle disease virus, avian adenovirus, they are inoculated in allantoic. So allantoic cavity. So there are different sites of inoculation in the embryonated egg. Okay, and different viruses can be grown likewise. Now tissue culture. So there are different types of tissue culture. One is the organ culture. The small bits of organ are used for the culture. Example is the tracheal rings for coronavirus. Another one is cell culture. That it has also different types, such as the primary cell culture, human amnion, chick embryo, monkey kidney. These are the examples of primary cell culture. Then these cells are obtained, the primary cell cultures, they are obtained from the freshly killed animals. Okay, and can only be passaged one or twice. Okay, and they are used for isolation of viruses, and primary isolation of viruses, and cultivation of viruses for vaccine production. These all terms, these all things may be new to you. Okay, when you go to the different systems, okay, different viruses, then after you will come to know in detail. Okay, it will be easier for you to again memorize it. Okay? So another one is the diploid cell culture, that is the semi-continuous cell culture, and examples are human embryonic lung cell strain, rhesus embryo cell strain. It can be passed up to 50 times, that culture can be done 50 times. Then is the continuous cell lines. These are the immortalized cell lines and may be passed indefinitely. They are made from the cancer cells in the human. Okay, they can proliferate indefinitely. That's why they are used. So examples are the Miller cells, that is obtained from the human carcinoma cervix. Egg cell, that is human embryonic kidney cell line. Now, whether it will inoculate the viruses in different methods, animal inoculation, cell culture, okay. So, whether the virus has grown or not, so that can be detected by different methods. Right? There are different methods. There are different changes, psychological changes that can predict that whether the viruses has grown or not. Okay? So, one of them is the cytopathic effect. There is cytopathic effect mechanismally. There is syncytium formation. That means there will be the fusion of giant cells, okay, in monolayer. Okay, fusion of the most cells and they form the giant cells. Okay, that occurs. The common example is the meiosis. Another one is the cell necrosis or lysis. The infected cell become pigmented and granular. So these are the effects that are seen after the growth of viruses in the cell culture. Okay. And cellular clumping, they do not fuse but produce the large clumps. The inclusion bodies. Okay. Inclusion bodies, they are elementary bodies. They are produced either in the cytoplasm or nucleus of the cells. They are specific in the specific in the different viruses. So these are the examples. If you are asked to MCQ that Cauchy type A bodies are seen in, then have to say the herpes virus. Okay, so these are the examples, some few examples. There are lots of inclusion bodies named. Okay, so these are the few examples of inclusion bodies that are seen in the cells. Okay, so first one is the veneri bodies that is seen in the pox virus. Next one is the negri bodies that is seen in the rabies virus. So these are the pictures showing the different inclusion bodies in the different viruses. Coli type A bodies, they are seen in the herpes virus. Okay, so at least remember few inclusion bodies name. Okay, that is the negri body, the most common that is seen in the rabies virus. Okay. Now, the serology. Serology is the most commonly performed diagnostic test for viral infections. This is the mainstay of viral diagnosis. This happens in primary humoral immune response to antigen and following the exposure to the virus, the first antibody to appear is the immunoglobulin M. Okay, and which is followed by the immunoglobulin G. Okay, the first immunoglobulin that appears after exposure to the pathogen is the immunoglobulin M, and then immunoglobulin G. Okay, which immunoglobulin is transferred from mother to fetus? So in cases of reinfection, the level of specific immunoglobulin either remains the same or rises slightly, but the immunoglobulin G is shown rapidly. Okay, I am showing the picture. So this is after the first exposure. The picture showing the rise of immunoglobulin, that is immunoglobulin M and G, but in the secondary exposure, after re-exposure, the immunoglobulin G has higher titer than the immunoglobulin M. Now, the serological tests, they are performed on the basis of on the principle of immunochromatography and ELISA. These are the most two common methods. So, immunochromatography. Have you ever seen that? They give us a So, in the next unit 1, that is immunology and blood, there you will see that they will learn about the antibody, antigen-antibody reaction, and they will have the database on antigen-antibody reactions. We come to at that time and we will see the different tests there. So, immunochromatography, it is also called as the lateral flow assay. Immunochromatographic assay or lateral flow assay, that is the most commonly used assay serological test for the viral diagnosis. Okay, so by this method we can detect either antigen or antibody. Okay, so antigen detection can be done for hepatitis B, that is hepatitis B surface antigen, coronavirus antigen, and dengue NS1, that is non-structural protein. Okay, so these are the antigen detection tests. Antibody detection for HIV by determine and SCV or tridode or any of the commercial case you can take. So By this method, we can detect both either the antigen or antibody. So how it works? They are found either in the cassette form or in the strip form. Okay. So this is the cassette form. Okay, and or it may be found in the strip form. Okay. What happens there? Whether if let's see the example of HBsAg, that is the hepatitis B surface antigen. Okay? So if you need to detect this, that means there should be the protein of antibody against this antigen. Okay? Usually the equivalent serological test result that is based upon the antigen antibody reaction. Okay? If you need to detect antibody, there should be in the test there should be coating of antigen. Okay? If you need to detect antigen, there should be coating of antibody. Okay. So here you see this is the sample pad. Okay, there are different compartments. Okay, so this is the collagen hole that is conjugated with labeled antibody against this HBsAg. Okay, and then after when you add patient sample, there is already antibody coated here. Now when you add patient sample and if the patient has the hepatitis infection, that means the patient contains the HBsAg. This antigen. Okay, then after they will travel through this strip, okay, by capillary action and then combine with this antibody. Okay, see there, this is the antibody, this is the antigen. Okay, the white shape is antibody and the round one is the antigen. Okay, so they combine together and that is detected here. टेस्ट यहाँ देखिरहेछ नि होइन पोजिटिभ टेस्ट यो के हो कन्ट्रोल हो यो टेस्ट हो ओके कन्ट्रोल भनेको एउटा गाडी टेस्ट गाडी छ कि छैन भन्ने कुरा जनाउनलाई हो द्याट सुड अलवेज कम पोजिटिभ ओके एन्ड देन दिस वान इज द टेस्ट लाइन this means the patient has hepatitis infection hepatitis B infection okay but here only the control line that means the patient has no infection. Okay, so we can detect both antigen and antibody. They are available as gaseous form or strip form. If you need to detect antibody, antigen is coated there. If you need to detect antigen, antibody is coated. Okay, so this is the basic thing. Now another one is the enzyme-linked immunosorbate assay. The name itself says it uses enzyme. Okay, and immunosorbate. Okay, that enzyme it can be used for detection of both antigen or antibody. Okay, and it is a cassette form or strip form market. But here it is a plate-based assay technique designed for detecting and quantifying. We can detect or quantify either antibody, antigen or different proteins, okay, and hormones too, right? So, enzyme it reacts with substrate to produce the colored product and it is seen like this, okay? So, these colored products they indicate the positive result, okay? So, there are different types of colored direct, indirect, sandwich, competitive and so on. There are different types of ELISA. Now, here is also the same basic principle. That is, if you need to detect antibody, then antigen is coated in the well. Okay, so these are the plates. It is plate-based assay. So these are the wells. Here they are coated with the antibody. If you need to detect antigen, okay, they are already pre-coated. See this example. I am showing the picture. So, ELISA procedure, how it occurs, how we do it. First, suppose this is the well. This is the single well over here. Okay. Now, it shows the coating of antigen in the well. Okay, okay. These are the antigens. Now, if you add patient serum and if there is presence of antibodies, that is there is infection, I know. Then after it will bind. Okay, yeah, bind coil, and after adding the patient serum, after adding the different enzymes and substrates, in each and every step their washing is done. Okay, so that the unbound antigen or antibody or substrate they all get washed out. Okay, so now see, here the antigen and antibody complex has been formed. Then after we add the enzyme that is labeled with anti-human immunoglobulin G. Now this anti-human immunoglobulin G, they are produced in different animals. Most commonly is the goat. Okay, so in goat the anti-human immunoglobulin are produced and they are tagged with the enzyme. That okay, so there are different types of enzyme. Okay, so that can be tagged with the immunoglobulin, and then when it is added and then after incubating and then washed, then after we add the substrate. Okay, and when this enzyme combined with substrate, then they produce the color that is detected. Okay, so this is the basic principle of the ELISA. Now, so this is all about today's class. For diagnosis, quality of patient specimens and their transport to the lab is very important. Okay, refrigerate all specimen until tested, but it is only for the viruses, right? And then after approach for the detection of viruses is the direct detection of virus by microscopy. Then after virus culture and different serological tests for diagnosis. So this is all about this class. If you have any question, you can ask me. What are the inclusion bodies? Rabies, okay, and other one, vulnerable bodies in pox virus,
virus, negri bodies, rabies,
the book, there are different types of inclusion bodies, and these inclusion bodies they are produced either in the cytoplasm or nucleus of the cells. Okay, they have different location, and that is the indication for the viral growth in cell culture. Okay, बुझे? Okay, so direct detection of virus is done by electron microscopy. Also, okay, also even electron microscopy. Okay, so what are can this virus? So can you grow these viruses in culture media? No, they are cell culture. Ordinary routine media में कर सकते हैं ना, but they can be used in cell culture है ना. Okay, so what are the examples? यही ना याद रखो, कम से कम नाम से याद रखो करो, है ना. एटलीस्ट टीचर नाम से याद रखो करो. बाइबल में सूर्य में क्या है? Okay. Actually, they are broad meaning. You can see the examples. Okay. Okay. So, what are the other method of virus detection? Serological test that is based on antigen antibody reaction. Okay, which type of antigen antibody reaction? Immunochromatogram and ELISA.
ELISA.
What is the full form of ELISA? Okay, so the description itself in the name. I know it uses enzyme, it uses immunosorbent particle. I know that is coated with the antigen or antibody. Right? Enzyme will react with the substrate and the color product will be visualized by ELISA reader. The ELISA reader will where detect. Okay. So what are the things that can be detected in ELISA? Antibody, or protein, hormones, complex hormones. Okay, but here we are concerned about the antigen and antibody. Okay, so those are our viral lab diagnosis of viral infection, multiple sclerosis, sir. Yes. Okay. It's all about today's class.
छत्ती निंद्रा बड़ी आज मेरी तेरे होटल लिखी
45, 49, 40,
तेरे होटल लिखी
41, 42, 43, 44, 45, 46, 47, 48, 49, 50, 51, 52, 53, 54, 55, 56, 57, 58, 59, 60, 61, 62, 63, 64, 65, दिवस के बाद 66, 67, 68, 69, 70, 71, 72, 73, 74, 75, 76, 77, 78, 79, 78, 79, 80, 81, 82, 83, 84, 85, 86, 87, 88, 89, 90, 91, 92, Yusuf Kumar Yadav, 93, 94, 95, present 96, 97, 98, 99, 2600, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34, 35, 36, 37, 38, 20 seconds. Roll number 11 to 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34, 35, 36, 37, 38, 39, 40, 41, 42, 43, 44,